Welcome to this tutorial introducing PHI-base, the Pathogen Host Interaction Database. My name is Alistair Irvine and I'm a software developer based at Rothamsted Research, where PHI-base has been developed. In this short video, I'm going to introduce you to PHI-base and show you some simple examples of how to search for data using the PHI-base website. For this demonstration, we will be using a new website that is being launched with a new version of the database. A test version is currently available with this website, shown in the URL at the top of the screen. PHI-base contains manually curated data regarding the effect pathogen genes have on the phenotype of a tested host. PHI-base covers a broad scope of pathogen and host species. Here you can see the figures for the current release of PHI-base. This shows over 4,000 interactions from 166 pathogen species infecting 110 hosts, causing 181 diseases. The data has been manually curated from over 1,200 peer-reviewed papers. So now we'll just try a quick search. So go to the top and click the search button, and that will bring you to the main search site. You can use the search box to perform a search based on any field in the database. For example, the name of a pathogen gene, a disease, or a host species. For example, if I want to search for interactions relevant to the BTP1 gene, I simply type BTP1 and click search. So here you can see that this gene has produced 4 interactions. In the list, we are shown the gene name, the phenotype, the species of the pathogen, the host species, and the disease. So we can see that in this case the gene was tested in 4 different hosts and produced a different phenotype in the string bean host compared with the other 3. To look at an individual interaction in more detail, click the plus. This shows a series of tabs giving details for different aspects of the interaction. So, in the gene tab, we can see the gene name and function, as well as a protein ID, which in this example is a UniProt accession. Clicking on the link brings up a new window with the details of this entry in UniProt. For a second example, I can search for all of the interactions with the barley host simply by typing barley and clicking search. Here we have a long list of interactions, and if we scroll down to the bottom of the screen, we can see that there's a total of 218 interactions, with the first 45 displayed on this page. But I may not be interested in all of these. On the left, there is the option to filter the results based on any of the listed fields. So, for example, I may only be interested in barley leaf scald disease. So, I can select the barley leaf scald disease and then click Refine Search. After we have done this filtering, you can see that the known interactions for this disease all come from a single pathogen species. If you wish to search on a specific criteria or a combination of specific criteria, you may prefer to use the advanced search. If we click advanced search, then we come to a page where you can specify the criteria for individual fields. For example, if we wanted to find all of the pathogens associated with a particular pathogen species, we simply go to the pathogen field and type in the species we are interested in. Click Go, and the results will appear for that particular species. So here are the results from the search. As you can see, the advanced search criteria is shown in the search box, and the interactions for this pathogen are displayed. As with the basic search, you can further filter the results using options on the left hand side. There's lots of additional information available on the website. The About Us page gives general information about PHI-base, listing our publications as well as papers that have cited PHI-base in their studies. If you are interested in analyzing all of the data in PHI-base, you can download the entire contents of the database in XML format or get the amino acid sequences in FASTA format, both available from the download menu. Note that you will be required to enter your details in order to use this service. Finally, if you do have any queries or suggestions, we'd be happy to hear from you at this email address. Also, if you want to keep in touch with the latest developments in PHI-base, you can subscribe to our PHI-base users mailing list following this link. Thank you for your interest in PHI-base, and I hope this has been a useful introduction for you.